Welcome back to another Sleepy Steve story. Before we drift off tonight, remember to subscribe to help Steve-O out, and I will keep making more sleepy stories to help you study gently, revise quietly, and settle your mind at the end of the day. Tonight, we are moving gently into one of the central ideas in biology: how information stored in DNA can become a working protein, and how those proteins help cells function, survive, communicate, and respond. We are in VCE Biology, Unit 3, Area of Study 1. In the last episode, we settled into nucleic acids, DNA, RNA, nucleotides, bases, genes, genomes, and DNA replication. Tonight, we take the next quiet step. We ask how a gene can be used. We ask how the sequence of bases in DNA can be expressed as a protein, and we ask why that process matters so much for living things. So breathe in slowly and let the idea become simple. DNA is information. Protein synthesis is one way that information is used. A gene is a length of DNA with a particular base sequence. That sequence is not random. It contains information that can be used to make a functional product, often a protein or part of a protein. Proteins then do many of the jobs of the cell. They can form structures, they can work as enzymes, they can act as hormones, they can sit in membranes, they can transport substances, they can receive signals. They can help cells divide, respond, repair, and regulate. This is why gene expression matters. A gene that is simply sitting in the DNA is like a recipe in a closed book. It exists, but it is not yet producing anything. When the gene is expressed, the information is read and used. A product is made. The cell changes what it is doing. In a calm exam sentence, you might say, gene expression is the process by which information in a gene is used to produce a functional product, such as a protein, that can influence the structure or function of a cell. Now we can move into the two main stages of protein synthesis. The first stage is transcription. The second stage is translation. Transcription happens first. It occurs in the nucleus of eukaryotic cells because that is where the DNA is located. The purpose of transcription is to make an RNA copy of a gene. More specifically, it produces a messenger RNA molecule, usually called mRNA. This makes sense when you remember that DNA stays in the nucleus. The DNA does not leave to travel to the ribosome. Instead, a copy of the relevant information is made in the form of mRNA. That mRNA can then move out of the nucleus and carry the message to a ribosome, where translation can occur. Let us walk through transcription slowly. A particular gene is selected. The DNA in that region unwinds and unzips. The hydrogen bonds between complementary bases break, exposing the bases on the DNA strands. One strand of DNA acts as the template strand. RNA nucleotides line up next to the exposed DNA template according to complementary base pairing rules. In RNA, the sugar is ribose rather than deoxyribose. RNA is usually single-stranded, and instead of thymine, RNA uses uracil. So, when the DNA template has adenine, the RNA nucleotide that pairs with it is uracil. When the DNA template has thymine, the RNA nucleotide that pairs with it is adenine. Cytosine pairs with guanine, and guanine pairs with cytosine. An enzyme called RNA polymerase helps join the RNA nucleotides together, forming a growing mRNA strand. When transcription is complete, the mRNA strand separates from the DNA, and the DNA strands rejoin. So, the result of transcription is an mRNA molecule with a sequence of bases that is complementary to the DNA template strand. It is useful to hold this in a very clear form. DNA is transcribed into mRNA. The base sequence of mRNA reflects the information in the gene. The mRNA then carries that information to a ribosome. In eukaryotic cells, the first RNA transcript may be processed before it is translated. Non-coding sections may be removed, and coding sections may be joined together. You may hear these described as introns and exons. Introns are removed. Exons remain in the mature mRNA. The mature mRNA can then leave the nucleus through a nuclear pore. You do not need to make this feel more complicated than it is. The main idea is that the mRNA becomes a usable message. Now we drift to translation. Translation occurs at ribosomes, either free in the cytosol or attached to rough endoplasmic reticulum. The purpose of translation is to use the sequence of bases in mRNA to assemble a chain of amino acids. That chain is called a polypeptide. The polypeptide may then fold and sometimes be modified into a functional protein. The key language here is the genetic code. The mRNA is read in groups of 3 bases. Each group of 3 bases is called a codon. A codon corresponds to a particular amino acid or to a start or stop signal. For example, a codon does not represent a whole protein. It represents one amino acid or a signal in the reading process. Amino acids are the building blocks of proteins. Different proteins have different amino acid sequences. The order of amino acids matters because it affects how the polypeptide folds, and folding affects the shape and function of the final protein. Now, how does the ribosome know which amino acid to add? This is where transfer RNA or tRNA becomes important. A tRNA molecule carries a specific amino acid. It also has a sequence of 3 bases called an anticodon. The anticodon is complementary to a codon on the mRNA. When the anticodon of a tRNA pairs with the codon on the mRNA, the correct amino acid is brought into place. The ribosome moves along the mRNA. Codon by codon, tRNA, molecules bring amino acids. The amino acids are joined by peptide bonds. The chain grows. Eventually, the ribosome reaches a stop codon. Translation ends. The polypeptide is released. So, in a clear sequence, the mRNA attaches to a ribosome. The ribosome reads the mRNA codons. tRNA molecules bring specific amino acids. Anticodons pair with codons. Amino acids are joined into a polypeptide. The polypeptide folds into a protein. That is the quiet rhythm of translation. If you are asked to distinguish transcription from translation, keep the comparison simple. Transcription produces mRNA from a DNA template. Translation produces a polypeptide using the mRNA sequence. Transcription occurs in the nucleus of eukaryotic cells. Translation occurs at ribosomes. Transcription uses RNA nucleotides. Translation uses amino acids. Transcription copies the message. Translation builds the protein. Let that settle for a moment. Copies and messages, codons and amino acids, ribosomes, and proteins. Now we need to connect protein synthesis to gene expression. Gene expression is not just the existence of a gene. Most cells in a multicellular organism contain the same DNA. A skin cell and a muscle cell can have the same genome, but they do not use all the same genes at the same time. Different genes are expressed in different cells at different times and in different amounts. This is one reason specialized cells can have different structures and functions. A cell may express genes that allow it to produce proteins suited to its role. A pancreatic beta cell can express genes involved in insulin production. A red blood cell, while developing, expresses genes associated with hemoglobin production. A muscle cell expresses proteins involved in contraction. The DNA provides the potential. Gene expression determines which parts of that potential are used. Regulation of gene expression means controlling whether a gene is expressed, when it is expressed, and how much product is produced. This regulation can happen at different stages. A cell may control whether transcription occurs. It may influence RNA processing. It may affect whether mRNA is translated. It may modify proteins after translation. For VCE purposes, it is enough to understand that gene expression is regulated, and that regulation allows cells to respond to internal and external signals. For example, a cell may increase production of an enzyme when a particular substrate is present. A cell may reduce production of a protein when it is no longer needed. During development, some genes are switched on and others are switched off, allowing cells to differentiate and form different tissues. The phrase switched on or switched off can be helpful, but remember that it is a simplified phrase. More accurately, gene expression can be increased, decreased, activated, or inhibited. Now, let us make the link between base sequence and protein structure. The sequence of bases in DNA determines the sequence of bases in mRNA. The sequence of codons in mRNA determines the sequence of amino acids in the polypeptide. The sequence of amino acids influences the folding, shape, and function of the protein. This is one of the most useful chains of reasoning in biology. DNA base sequence affects mRNA codon sequence. mRNA codon sequence affects amino acid sequence. Amino acid sequence affects protein shape. Protein shape affects protein function. Protein function affects cell activity. Cell activity can affect traits, health, and survival. This chain becomes especially important when we later discuss mutations. A mutation is a change in the DNA base sequence. If a mutation changes the mRNA codon sequence, it may change the amino acid sequence. If the amino acid sequence changes, the protein may fold differently. If the protein folds differently, its function may be altered. Not every mutation changes protein function. Some changes are silent, some are minor, some are significant. But the pathway from DNA to protein gives you a way to explain why genetic changes can matter. Now we can practice the exam language gently. If asked to explain how DNA directs protein synthesis, you could say, a gene contains a specific sequence of DNA bases. During transcription, an mRNA copy is produced using the DNA template strand and complementary base pairing. The mRNA moves to a ribosome, where it is translated. The ribosome reads the mRNA in codons. and tRNA molecules bring corresponding amino acids. The amino acids are joined into a polypeptide, which folds to form a functional protein. That answer has the key terms. It has the order. It has cause and effect. If asked to explain the role of mRNA, you could say, mRNA carries a copy of the genetic information from a gene to a ribosome. Its codon sequence determines the order in which amino acids are joined during translation. If asked to explain the role of tRNA, you could say, tRNA carries specific amino acids to the ribosome. Its anticodon pairs with a complementary codon on mRNA, helping ensure the correct amino acid is added to the growing polypeptide. If asked to explain the role of the ribosome, you could say, the ribosome is the site of translation. It reads mRNA codons and helps join amino acids together to form a polypeptide. Now we can slow down again. Imagine a cell at night, not as a machine roaring with noise, but as a quiet library. DNA rests in the nucleus like shelves of instructions. Not every book is open. Not every page is read. But when the cell needs a particular protein, a gene is transcribed. A message is made. The message travels. A ribosome reads it. Amino acids arrive. A chain forms. The chain folds. And somewhere in the cell, a new function becomes possible. That is the beauty of protein synthesis. It turns stored information into biological action. Now let us include a few common traps. First, DNA is not directly translated. DNA is transcribed into mRNA. mRNA is translated into a polypeptide. Second, codons are found on mRNA, while anticodons are found on tRNA. Third, a protein is not simply a string of DNA bases. A protein is made from amino acids. Fourth, transcription and translation are separate processes with different locations and different products. Fifth, gene expression can be regulated. Cells do not express every gene all the time. These small distinctions can protect marks in short answer questions. Now we can revisit the key terms one more time, slowly. A gene is a segment of DNA that contains information for a functional product. Transcription is the process of producing mRNA from a DNA template. mRNA is messenger RNA. It carries the genetic message to a ribosome. A codon is a sequence of 3 bases on mRNA. tRNA is transfer RNA. It carries an amino acid and has an anticodon. An anticodon is a 3-base sequence on tRNA that pairs with a complementary mRNA codon. Translation is the process of using mRNA to assemble a polypeptide. A polypeptide is a chain of amino acids. A protein is a functional molecule formed from one or more polypeptides that fold into a specific shape. Gene expression is the use of genetic information to produce a functional product. Regulation of gene expression is the control of when, where, and how much a gene is expressed. Now, let us finish with a calm, full summary. DNA stores genetic information in the sequence of its bases. A gene can be transcribed to produce mRNA. During transcription, RNA polymerase helps build a complementary mRNA strand from a DNA template. In eukaryotic cells, the mRNA may be processed before leaving the nucleus. The mRNA then travels to a ribosome. During translation, the ribosome reads the mRNA in codons. tRNA molecules bring amino acids using anticodons to match the correct codons. The amino acids are joined together in a particular order, forming a polypeptide. The polypeptide folds into a protein. The protein then contributes to the structure or function of the cell. And underneath all of that is the larger idea. Cells use information. They regulate information. They turn information into molecules, and those molecules help create life. So tonight, as you let the details soften, hold on to the pathway. DNA to mRNA, mRNA to amino acids, amino acids to polypeptide, polypeptide to protein, protein to function. That is protein synthesis, that is gene expression, and that is enough for now. Sleep well and let the cell keep working quietly in the background.